Yeah, so it's my pleasure to introduce Dr. Jason Flannick, who's an assistant professor at Harvard Medical School and an associate member of the program in medical and population genetics at the Broad Institute. He's also a colleague, friend, and close collaborator of mine for many years. Jason did not originally begin in genetics, actually. Was a quantitative scientist majoring in physics at Stanford and then getting a PhD in computer science. And then he came to the Broad Institute to be a postdoc in statistical genetics with David Altshuler. And that's where we both met as postdocs. I believe Jason taught me my first set of Unix commands, and that was many years ago now. So I'm always grateful to him. Dr. Flannick's lab works on type 2 diabetes, genetic variation, and he'll talk about that in his talk. But what I want to highlight is a contribution that Jason has made to science that sometimes goes underappreciated, and so which is the aggregation, harmonization, and building pipelines on genetic data to make it accessible to people. So Jason was one of the pioneers in democratizing genetic data so that you didn't have to know how to code or have compute clusters to actually be able to understand and look at genetic variation in a gene. And I think this has spurred an incredible amount of research and changed some of the ways that we do science. And so without further ado, take it away, Jason.
All right, thank you, Amit, for the great introduction. And welcome or hello to everyone from New England. I do wish I was in the warmer weather, given the cold front we're having. I do want to apologize to those who read this pamphlet and were expecting a talk on making human genetic data. That is a very interesting topic, but unfortunately one that I don't think I can actually talk about. The actual topic is going to be making human genetic data more useful, and this touches on what Amit just said. So this is the focal point of our lab. If you're not familiar with us, we're based out of Boston Children's Hospital and the Broad Institute, where we work on computational methods for integrating and analyzing genetic data, particular interest in rare coding variants in type 2 diabetes. And this research grew out, ironically, out of a very unrewarding experience I had as a postdoc. And to tell that story, back in 2015, it was the beginning of the Accelerating Medicines Partnership, or AMP, for type 2 diabetes. And this was like a partnership of 5 pharmaceutical companies and the government and big international consortium of geneticists who we were trying to figure out how to make human genetic data more relevant to drug discovery for type 2 diabetes and related traits. And one of the first analyses that we were asked to do by our pharma partners was to look up in all of the data we had collected at the time and see if we could identify or prioritize potential novel diabetes drug targets. So I was an enterprising postdoc at the time, and so I spent years actually working on mining through all of our data, developing new analytical methods and talking to everyone I could, and ultimately produced this 100-page slide deck on all these targets I thought were interesting, even longer appendix. And I sent it out to all of our partners and was really excited to hear what they would say. But what I heard back from them was actually absolutely nothing. Not a single person contacted me about any of the genes, and I was really excited about them. And at the time I wrote it off as kind of, okay, genetics is kind of a niche. Maybe they're not really interested in genetics. Maybe I didn't present it correctly. But a year later, I was at visiting one of our partners. And in a one-on-one meeting, he pulled me aside and showed me this internal white paper where he had actually gone through every single gene that I had given him. And he had written up like 5 pages on it. And he had ruled out every single one as a potential drug target immediately. And so what this meant was that it wasn't that like he wasn't interested in genetics. It wasn't that the data I had sent him he didn't understand. What it was is that the data I sent him weren't useful to him. And so that's really the question that, I've really thought about ever since then, and, about what we can do about it. And I'm, trying to understand more about how we've approached that question. I think it's useful to go back a little bit further to understand what type of data we're talking about. And also, I should have said at the start, I'll pause through at several points today to ask for questions, but please put any into the chat as we go. I will look at that and probably answer some of those as we go or at the stopping point. So please, just any question that comes up, just pop it into the chat. Okay, so to now just think about what these data were. Okay, so the story for that begins a little earlier, back in 2008, when everybody was talking about the case of the missing heritability. So what this was something that came out of the first GWAS. So if you're just a quick primer what GWAS are. So a genetic association study is when you take people with and without a disease or people who vary on some trait, and then you look for variants that differ in frequency among people with and without disease. And in principle, what that does is points you to a region of the genome that you can then study further to get insights into the genes and processes that underlie disease. And this is a very rigorous and unbiased way of doing this. And genome-wide association studies are a particular type of association study where you focus only on common variants. And this is primarily because it's very inexpensive to do these, because you can genotype a small number of them, or a relatively small number of them, and then through correlations among nearby variants, indirectly study all of the variants, all of the common variants in an individual. So the first GWAS had been published, I think, in 2007 for diabetes and related traits, and they were successful at identifying some loci, which was by no means preordained, but there was quickly a backlash that focused instead on the limited proportion of heritability or genetic basis of disease that they explained. And in particular, there was one very vocal group of people who argued that the problem was that we shouldn't be looking at common variants, that rare variants were going to be much more valuable for understanding disease. And so the argument was that clearly these common variants have very weak effect. They're going to be spread, if they're going to explain disease, they're going to be spread across thousands of genes and won't tell you anything. But these rare variants might have much larger effect, and therefore explain a lot of heritability on the one hand, or on the other hand, directly point you at a gene with a very large effect, and the rare variant would tell you what was being done to that gene to produce the effect. And that this was, would lead to much more direct therapeutic insights, as had been done for PCSK9 a couple years earlier. So our group, I was a postdoc at the time, we were studying this for type 2 diabetes, which I'm sure you're all familiar with. For the purpose of today, it's enough to know that it's a very representative complex disease. So genetically, it looks a lot like a lot of other complex traits. And then pathophysiologically, it has two major dysfunctions: insulin resistance in peripheral tissues like liver, muscle, and fat, impaired insulin secretion from the pancreas that together means that people present with hyperglycemia or elevated blood glucose levels that has some significant public health consequences. So we were looking at rare variants relevant to diabetes. And initially, we couldn't afford to sequence the whole genome or exome of a person. So we looked at genes that were involved, that were implicated from diabetes GWAS loci. In particular, SLC38, had been identified from an early GWAS and had a very well-known cellular role. And there was a very clear hypothesis that inactivating it should increase risk of diabetes. But the mouse and cell models done at the time had really failed to establish that conclusively. So one of our earliest findings was 12 loss of function mutations that we discovered in 150,000 people that we sequenced. And so in principle, these are people who have 50% reduction in activity of this gene. And by looking at whether or not they are predisposed to diabetes, you can actually establish whether once and for all inactivation or activation of this gene is risk increasing. So just one side note, which will be relevant for the remainder of the talk: when you analyze rare variants, you can't analyze them like a GWAS because you don't have enough power to tell frequency differences for individual variants. So you typically pool them and then look at aggregate frequency differences. And these are called burden tests or aggregate tests. And every time I talk about rare variant associations today, I'll be referring to these gene level rare variant associations, which are really combined effects of many variants within a gene. Okay, so when that's applied to these 12 loss of functions, you get in SLC38, you get a protective signal. So in fact, it was the opposite of the previous models people were working under. And what that was initially somewhat controversial, but over the years has since been replicated as we've collected more data. And I think, I don't know, last year or 2 years ago, cell and more modern cell and mouse models started to unpack exactly how variation in this gene is impacting diabetes risk. Now, so that's like a great example, right, of what these high impact rare variants are supposed to find. But unfortunately, when we expanded beyond these candidate genes, and at the time, this was a ton of genetic data, we unfortunately didn't find any more. We didn't find any, in fact. And instead, we actually collected data that was consistent with this common variant model of many variants with weak effects spread across the genome that is what's responsible for the bulk of diabetes risk. And that's been true, I think, for most complex traits since then. So that's what's led to this explosion. What happened was these rare variant models kind of fell by the wayside, and instead the field shifted towards larger and larger GWAS, which if you've been following the field, you've just seen this absolute explosion. This was from a review we wrote in 2016, and you can see these early GWAS down here in 2008. At the time, this was like a ludicrously large sample size in 150,000 people. But even that, if you go out, in the 4 years since, is just paled in comparison. So you have these GWAS, they're just cranking out hundreds and thousands of associations for diabetes and related traits. They have this one issue, which is that the very correlations that make them cheap make it hard to take an association and link it to a gene. So an association, you get this peak like this. And so there's like one causal variant, but many associations. And because they're in non-coding regions of the genome, it's not clear what gene they're implicating all the time. And so most of the field, I would say, has shifted towards developing new data collection techniques and methods to try to figure out how you can at scale translate these associations to function. And there's actually a ton of great work being done at UCSD to do this, particularly in the profiling of the epigenome and non-coding variants here. So that's one data type we're dealing with, right? GWAS, tons of data, high power, but limited in terms of what resolution we have for causal genes. Now, meanwhile, you have these rare variants, which in principle complement GWAS very well, right? They give you a direct link between a gene and disease. And as we saw in the case of SC38, they can sometimes actually give you the directional relationship, whether lowering or increasing activity is what increases risk. The slight problem is that we can't seem to find any for type 2 diabetes. Now, unfortunately, though, it's not that they don't exist, you just need more data to identify them. And we quadrupled sample sizes 3 years later and found for the first time significant exome-wide significant rare variant gene level associations. And more intriguingly, for what I'll talk about today, there was evidence that there were many more to be detected. So this is analysis that I'll show multiple times. You take a set of genes that you think might have some reason to be involved in diabetes. So this is like the gene is implicated in a different monogenic form of diabetes targeted by a diabetes drug. When you knock it out of a mouse, it gets something like that looks like diabetes. And these box plots show the strength of association for genes in that set, which shifted upwards relative to the expectation. And so what this model kind of suggests is that there's actually a lot of these rare variant associations spread across a lot of diabetes relevant genes. Heritability-wise, they don't explain a ton. So the red dots here are the heritability by these gene level signals compared to comparable common variant. And you can see it's far less. But it does give you this model whereby, and actually the model I work under is that pretty much any diabetes relevant gene will harbor some rare variants that are associated with diabetes. So I'll get back to why this is actually relevant to the question I posed at the start. But before I do that, I just want to describe a study that just came out of our lab last fall that provides more recent and further support for this model. And this was not for type 2 diabetes, but for a related quantitative trait. So the study I just described was called AMPT2D. And it was primarily for diabetes, but we also had other quantitative traits measured in the same samples. And a postdoc in our group worked on analyzing these together with, at the time, the UK first 50,000 exomes that had come out of UK Biobank. And when we did this, we found rare variant associations for a number of these traits, the number shown in gold here. Not nearly as many as the common variant we found, but more than we found for diabetes. Most of these were with lipid traits, which if you know anything about that field is not really, it's kind of old news. but there were a number associated with HbA1c, which was relevant to diabetes. So if you're not familiar, this is a biomarker that's commonly used to diagnose diabetes. It measures the fraction of hemoglobin in red blood cells that is glycated over the course of so it's a stable measure over the course of a month. And so you can measure without requiring people to fast. And typically you can use this as a diabetes diagnosis, usually 6.5% and above categorizes you as having diabetes. So we found 3 gene level associations with this trait, 2 of which, piezo1 and G6PD, are genes that are known to have roles in red blood cell function. Okay, so that's interesting because there was a GWAS for HbA1c that had been published several years earlier, where the main finding was a common variant in G6PD that was quite common in African Americans that had a quite large effect on reduced HbA1c. And this was categorized as an erythrocytic variant. And this was a variant that, based on associations with red blood cell traits, was thought to act through erythrocytic function, or red blood cell function, compared to the glycemic associations that had associations with other glycemic traits. And this was important because the glycemic variants predicted whether people would get diabetes, but the erythrocytic variants did not. So the model you can think about is that the erythrocytic variants are not actually predisposing you to diabetes or affecting whether you have hyperglycemia. They're somehow messing up the red blood cell function. And so they're messing up the measured A1C. So they're breaking the normal threshold that you should expect to see for type 2 diabetes. And what this means is that if you're an African American who carries this variant, you may be underdiagnosed because you might be over the threshold and have diabetes, but your measured A1C will be below the threshold. Okay, so this was like, a potentially interesting finding. And we had rare variants in these genes, which had pretty strong effects, and as stronger than you typically see for GWAS associations. So the obvious question was, do people carrying these variants, rare variants in these genes, also fall prey to some of these misdiagnoses? So to test that, we used our AMP-T2D exomes, for which we had A1C measurements, and we also had independent diabetes diagnoses based on fasting glucose measures, OGTT measures. And then we took variant carriers and we adjusted for the effects. So if they had a reducing variant, we would add that to their A1C. And then if they moved over or under the threshold, we would reclassify them, okay? And then we tested whether the reclassification was accurate using the independent measure we had, okay? So you can then quantify how many people are reclassified, and you can say, what's the proportion of those reclassified that is accurate, and how does that compare to the expected number based on matched people who don't have the variants? And then you get these plots that look like this. The size of this diamond is the number of people reclassified. The odds ratio is like a measure of accuracy enrichment. So this would say 6.5 times more correct reclassifications than expected by chance. So when you compare those 2 rare variant signals to the common variant score, you see that it pales in comparison. There's very few people, and it's not accurate. It's not statistically significant in terms of being able to identify true misdiagnosed cases. So we expanded the model using these same gene set associations I mentioned earlier, right? So we took gene sets that were implicated from mice in red blood cell function. And just like before, you see evidence that many of these genes have stronger than expected associations. And you see the same thing if you take gene sets that, based on mouse models, are involved in hyperglycemia. Now, interestingly, only the erythrocytic rare variant associations trend towards reduced A1C associations, which is consistent if they're impairing red blood cell function and therefore reducing the measured A1C. And so then the question is, can we now use these to augment the model that we have with just these 2 exome significant signals? So this was, I think, the first instance of actually trying to do this. And there's actually 3 challenges you have to solve. So you first need to identify genes that we think are truly associated with A1C. And for that, we tried some different criteria based on their p-values for A1C, as well as their mouse annotations. Then you have to filter those down to genes that we think act through erythrocytic pathways, which are based on red blood cell associations like the GWAS, as well as mouse annotations. And then you have to figure out for a variant in the gene, what is the predicted effect on A1C? And as I mentioned, rare variants, you can't estimate those individually. So what we did was we pooled them using that strategy I described, based on different measures of how damaging they were, estimated then the aggregate effect, and used the aggregate effect as a proxy for variance in that annotation class. So it has this nice property that you actually don't need to observe the variant directly. You can apply this to a variant in a test sample that's never been seen before. So we trained this model in the UK Biobank data. We tested it in the AMP-T2D genes data. And the resulting model had 22,000 variants in it. And as you can see from this box plot here, had now significantly more people reclassified and was now statistically accurate. And when you added it to the common variant score, since for the most part, these variants are independent, you get a substantially improved reclassification score if you're trying to find people who are misdiagnosed by raw A1C. Okay, so what's interesting though, is that if I told you rare variant score 10 years ago, you would think that this was primarily people who were walking around with like a variant that is rare and has like a huge effect on A1C. But in fact, because the variants are so rare and there are so many of them, if you plot the distributions of scores, which are shown in gray here, this is across different ancestries, it actually looks a lot like the common variant polygenic scores. So it's actually highly polygenic. It doesn't look at all like this high impact variant score. So it's actually kind of ironic that this model, I think, at least I really do believe that rare variants do play a large role in complex traits, but not at all in the way I think that these original models propose. They don't actually explain a ton of heritability, and they look a lot more like GWAS and being spread across, hundreds of traits, hundreds of genes. And I think that's kind of ironic. And I mean, that's, I guess, a glass half empty way of looking at it. I think the more glass half full way is that these data do in fact provide really complementary value to GWAS. And so this is the second data type that we were working with. And it also gives you signals across many genes. And so that brings me back now to the original question I proposed, which is why then, when we mined these data for our pharma partners, were they not useful? And so I guess I'm going to switch gears a little bit. So I guess I'll pause to see if there are any questions on that story before I go on. I don't think I see anything in the chat, but does anybody have any questions or on that?
Actually, I have a question.
Yes.
So the question is, so when you talk about polygenics, so there are two types of polygenic situations. One is that in the same gene, let's say P53 in cancer, right? So multiple positions, and you have technically different mutations, but their result is the same gene. So if you aggregate your signal per gene, right, or per protein, will it change the situation? Why do you insist on all different targets?
Oh no, that's a good question. So yeah, so 21,000 variants. I actually forget the number of genes. I think it's on the order of like 500 or 600 genes. So you do see both, right? You see a fairly large number of genes, which, there probably should be more if we had more power. So it's kind of polygenic in both ways, right? So you have a lot of genes, you have a lot of variants within the genes. And from the perspective of this plot, it doesn't really matter because I'm plotting people, their scores. But yeah, I mean, in principle, there is two levels of grouping here. And I don't know what implications that has for, thoughts about these models, but yes, that is an important point to raise.
Thank you.
Jason, I have a question as well.
Yeah.
I'm trying to put together that, we consider rare variants that are functional to be more likely to be high impact or have a larger effect on the gene function and hence biology. But your finding is that overall, their effect size distribution is similar to the common variants. So is it just that the distribution has wider tails in the rare variants, or how do you put those two together?
Yeah, so the way to think about this is individually, okay, the rare variants have much larger effects than an individual common variant. Okay, so I don't know I can't remember the ratio. I think it's like 3 or 4-fold. But if you look at a person, they carry many more common variants than they carry rare. So this is actually only a score of 22 common variants, but on average, a person will carry like 10 of them. And the sum of all those effects is more than the average sum of the rare variant effects in that person, if that makes sense. So there will be people at the tails of the rare, just like there will be people at the tails of the common. I don't know if we have enough data to actually opine on individually, like how fat the tails are, but it's actually, it's the same thing that we've seen time and time again, which is like most rare variants in the population are rare. Most variants in a person are common, right? Most heritability is due to common, larger effects are due to rare. So things look very different individually versus an aggregate. And that's really what you're seeing here. It's actually a very, it's in some sense is both obvious, but also interesting here. So I don't know if that made sense, but that's kind of what's happening. And I'll touch a little bit more on this at the last story at the end when we look at youth onset T2D.
Thanks.
All right, let me move on because I'll have time. I want to also have time for questions at the end. Okay, great. So yeah, please question in the chat. That would be great. Okay. All right. So back to this question. So we have all these signals. What's going on? So, okay, you may have guessed that, I kind of described data we had just published, a couple of years ago, but I sent the data to this collaborator in 2017, and maybe it's just about power, right? That we just didn't have the signals before. And to be honest, that probably should have been the reason, but it wasn't at all. Like there was no questioning of our collaborator of, whether the signals were accurate. What had happened was that he ruled them all out due to completely non-genetic reasons. Okay, so some of the genes were like already had a known mechanism. They had a drug on the market, not going to follow. Others had no known mechanism and just the cost of pursuing them was too high. And others were just expressed in tissues that made them complete no-goes. So in other words, they were all ruled out completely before any genetic study would have been done. Okay. So I think that actually illustrates a very fundamental divide between people generating genetic data and people who might use it that I think is underappreciated. So geneticists like myself, we operate under this, as I said, unbiased forward genetic approach, where we let the human population tell us where to focus. And then in principle, you then map that to biology. But now that we're starting to actually have to, practically do this, I think what we're seeing is most people doing that work, it's not like they have limited flexibility to go after any locus that will emerge, right? So in my pharma, they had very rigorous constraints on what genes they would pursue. And I think, I don't want to speak for academic labs, but the ones that I've worked with usually specialize in a cell type or a tissue. And they're really, while they might be interested in new findings, they're really not going to actively pursue those unless they can do so, with some of the tools they already have on hand. So this is the model that I think of as like, what can we take genetics instead to just throw new things over the wall? Can we try to, envision a person who did a differential expression screen and has 100 genes and there's some cost and benefit to pursuing them. And can we somehow take genetics and use that to help them make better decisions as to what to pursue in terms of the cost benefit calculus? And thinking about exactly what this means, like how do you calculate genetic support? What methods do you do? What data do you integrate? Can you do this based on not just the explicit genetic data, but other things, globally is really what we think about. And so the one issue is that I don't think most people either not interested in this or don't think about it. This is from an essay we wrote last year. As part of this, we examined papers from Nature, Science, and Cell over a 3-year period that had proposed diabetes, either drug targets or new genes from experimental systems. And in only 10% of the cases, papers was like genetics even mentioned, and nobody's looking up genetics to ever support any of these findings. And I think that that's like a huge opportunity, actually. Can we help get genetics more into these type of studies? And so, as Amit mentioned, the diabetes knowledge portal, which our group built as really as part of, that was the main product of this first phase of the Accelerating Medicines Partnership. The goal of that was to help get these data more easy to access and available to people, so they didn't have to use a postdoc to do their gene screens. They could just type them into a browser on their own. So this is basically a resource of GWAS data, but it's integrated with a lot of epigenomic and functional genomic data that's actually stored here, I guess, or there at UCSD in Kyle Galton's lab. And what we do is we don't just, we try to process this through a series of methods and integrate it in ways that allow more simple queries than just downloading the raw results. And so let me just give you an example of what I mean by that. Like the simplest query you could ever consider doing would be to just look up the association of a SNP with diabetes, right? But it's actually non-trivial when you have 40 GWAS for type 2 diabetes, right? Because you have differential evidence. These are the p-values, like these are just hypothetical, right? But if you think about, well, let's report the lowest p-value, that's completely unprincipled, right? But actually is what everyone does if you're looking up something in the literature, or if you're a resource that's just putting out associations that are significant in any data set. If you take the largest data set, that's more principled, but you throw out data. And if you meta-analyze them, that doesn't work if there's any overlap between the studies, right? So as part of Mike Benke's group and Gonzalo Abacase's group at Michigan have developed a method that infers overlap across studies without just based on the distribution of statistics, and allows you to do a meta-analysis adjusting for that. And when we've looked at the accuracy of these associations, they do replicate at a much higher rate in a leave one out analysis than do these other methods. So it's a more accurate way of combining them. And that's what the portal shows. And so as a result, the portal includes associations that are absent from the data sets. And as you would expect, this is like a plot of one of them. Each dot is a study. None of the p-values reach significance, which is this line, but collectively, when combined, they nudge you over the line. But we also remove associations that are in small data sets and not replicated in larger. And as a result, we hope, or our goal is to make the associations reported in this resource kind of the canonical set for diabetes. And so we capture most of those that are in other resources. I have two here. Most of the ones that we don't capture are not available publicly. So these are from studies that published their results but didn't make the data available. And the remaining ones are mostly in older and smaller studies, which are important, but again, not kind of a little outside of what we're trying to capture. And then the portal has these access mechanisms, a variety of them, which I won't have time to go through. Instead, I just kind of illustrate how they work with a paper that motivated that essay I described a couple of slides ago. So I read this paper in Cell a couple of years ago, and they had identified this new gene, SYN3A. I don't know if it's a new gene, but it was a novel interaction with FOXO1 in the liver. And they therefore proposed that this could be a pretty effective therapeutic target. And I thought, I wonder if there's a GWAS association nearby SYN3A. That would be kind of cool. So the paper didn't do it at all. But I looked it up in our portal, and this is what you get when you type in a gene. You get like a region, and then you get all the associations nearby. And there is a diabetes GWAS association nearby. And if you look up FOXO1, you go a little bit further, you look at the rare variant associations. There's a diabetes association. So it's not like genome-wide significant, but it's suggestive. And I thought, I wish they had included these data, because at least to me, that as a human geneticist, that would have made me more, convinced by their hypothesis. But, as I thought about it further, I'm not sure what they would have done, right? You have this, GWAS variant nearby SYN3A, but it's also nearby 5 other genes. You have this rare variant signal for FOXO1, but it's not exome-wide significant. Like, what does that actually mean, right? So what we tried to do in this essay was to borrow from, if you follow 538, to predict what they do is they do a great job of converting polling data into probabilities of victory. And what they're doing under the hood is that they have what's called a prior, which is how likely is each candidate to win based on fundamentals like the economy and incumbency status. And then they observe evidence, which is polling data in their case, and then they use that to update the prior to a posterior probability of victory. And this updating factor, the evidence is summarized in what's called a Bayes factor, which you don't need to know what that is. Essentially, the higher the Bayes factor, the more your prior gets shifted towards a positive outcome. So for genetics, what we want to do is we want to try to predict how likely a gene is to be involved in disease. That's kind of a vague construct. I can elaborate on it, but let's just assume that's a 1-0 outcome. The evidence is our association data, and we want to convert that to Bayes factors. So you do need a prior, and we have a whole project in the lab to figure out how to calculate priors for genes. For today, just assume you either have a kind of rough 5% prior that seems like the global number of order of magnitude genes involved in T2D. You could also have a more higher investigator defined prior, like if you have mouse data for it. But what you can then do is, in this essay, we outlined a series of simple rules based on previous GWAS publications that based on, first, is there a GWAS signal nearby the gene? And if so, is it nearest to the gene? Is it a coding variant? That we could map those to probabilities based on previous published estimates. And then an equation that was originally done for GWAS, but we could extend to rare variants to directly map the association signals to Bayes factors. And putting those together, you get this very crude, but I think at least slightly clearer table, which just look up how strong is the rare variant signal, how strong is the common variant signal, and then you can map that to a posterior probability under these conservative and optimistic scenarios. And I think when you do that for SYN3A, it turns out that it's something like a 90% probability if you believe in the mouse data, or like a 50% if you don't otherwise. So obviously we like to extend this to a more principled and comprehensive model. And again, we have a project in the lab to do that too. The idea is that you kind of model the gene's causality as this latent variable, and then you model the distribution of SNPs nearby, conditional upon that. You actually fit those distributions and parameterize them, given a lot of work that's been done in the field in the last 5 years, together with a lot of the epigenomic data we have and heritability estimates across the genome. And then you also have information about what annotations the gene has, like its expression pattern or mouse phenotypes. And those you can also fit based on observed data, which you can think of this as like a principal way to calculate the prior. And so we have a postdoc, Alex Llamas, in the lab working on this. And this is just what the actual model looks like, just given the common variant SNP data. You can see it's quite complicated, but in principle, totally fittable, and you can sample from it using like a basic Gibbs sampler that looks in our preliminary work to be working pretty well. And so the hope is that that will be plugged into the portal to support this workflow, where you can type in a gene, type in your phenotype of interest, and then get back this kind of intuitive score. So this is the score for SYN3A. This is using our current simplified model, and play around with different priors. Here's the two I showed. So I guess it was 70% for the 5% prior and 92% for the 20% prior. Just as an aside, obviously we grew this out for type 2 diabetes, but we've collaborated with numerous other disease communities, each one of which helps curate data sets for their trait of interest. And we've built a whole range of portals for querying those traits as well. And in October at ASHG, we launched, with the help of NHGRI, this association to function knowledge portal that brings them all together under one umbrella. So if you're interested in other traits besides diabetes, I encourage you to check that out at a2fkp.org. Okay, so I guess I'll pause one more time. I have one more story. It'll take about 15 minutes, so I have time for a couple questions if anyone has them on that part or anything to date. All right, so I'll continue. And so this is this is again, switching gears a little bit, but kind of follows from that, so the vision of this portal is that any trait studied in the population or complex trait, you can look up associations for. But as we've tried to apply that, in partnerships with people who are really studying a disease, and in particular, as you get to rarer diseases that we work with at Children's, you have a problem, which is that most of the traits people are interested in are not represented in that portal. And the reason for this is that all these GWAS model diabetes is like a 1-0 trait, like you either have it or you don't, right? But real people who have diabetes don't all look the same. This is a model from Mark McCarthy that's been criticized by implying you could just bucket people into different bins based on the pathways that are perturbed. But I think it's uncontroversial that people with diabetes will be heterogeneous and probably have different phenotype presentations. And I'm involved in this NIDDK funded project called Radiant that is recruiting patients with atypical forms of diabetes. And so those patients, obviously, diabetes is like an inaccurate description, right? There's all sorts of additional phenotypic information that we have on these people. And ideally, what you're trying to do is explain all of these phenotypes. That's the whole definition. And the more and more that you subdivide patients, or even if you're just a drug company trying to stratify patients for personalized treatment, the more and more everybody looks like they have a rare disease, like not necessarily like, super rare, but, disease phenotypes are probably fractal in the extent that you can subdivide until everybody's unique. And so for this project, as I think for most rare disease, we're doing this, filtering of variants according to these qualitative kind of ad hoc criteria. And we look through them all manually and try to do what, my old advisor kind of uncharitable, maybe a little unfairly called post hoc storytelling, where you try to just use, all sorts of literature searches and biological knowledge to explain the disease with a single variant. And this, is not usually successful in our project. And it's I think successful around 30% of the time for rare diseases. So it would be really nice, I think, if we could leverage all this statistical rigor and gigantic data we've collected for these common diseases to help in that process somehow, right? Now, of course, you don't have like this phenotype in our portal, but I think intuitively, if you just statistically wanted to rank genes, those that have T2D associations should probably be higher up the list, right? So this is from another review we wrote in 2016. These red arrows here are genes that are involved in monogenic forms of diabetes that also have mutations that cause late onset T2D, right? So there's clearly some genetic overlap. And the model that I work under is, if you think about diseases at the pathway level, a single mutation might cause a rare disease with a very particular phenotype, but mutations of weaker effect in the same gene or pathway might predispose to a complex disease that looks somewhat similar, but also resembles other diseases that might result from mutations in different pathways. And I think there's evidence for this. Like, we have multiple pathways we know are involved in type 2 diabetes that harbor both monogenic disease mutations for like neonatal diabetes or MODY, as well as late onset T2D mutations. And conversely, if you take diabetes GWAS associations and kind of profile their pattern of associations across other traits, you can bucket them into different clusters. And each cluster you can kind of map to different pathways and maybe even different patient subtypes. So I think this question that we're really exploring at Children's is, to what extent can we then use these kind of observations to transfer associations from common disease data to help somehow guide investigation of more specific phenotypes or even rare diseases. But the degree to which you can do that depends obviously on the degree to which phenotypic similarity predicts genetic similarity, which is completely unknown. And obviously won't hold in all cases, but the question is, to what degree does it hold? So I'll give one final study to provide maybe some more, clarity of that model for diabetes. Okay, so this is called the Prodigy study, and it's of type 2 diabetes in youth. Okay, so these are children, teenagers who do not have monogenic diabetes or they don't present like that, but they have type 2 diabetes 20 or 30 years earlier than people normally get it. It's a very interesting condition in its own right, because it's increasing rapidly in prevalence, particularly for underrepresented minorities. But it's also very interesting genetically, because it's kind of more extreme from type 2 diabetes, and it's not as extreme as a monogenic disorder. And so what's going on? And so there are these two registries, Search and Today, that have been collecting these patients for a while. You can see here their age of onset at about 13 for type 2 diabetes. Most of these children are obese, and so you could hypothesize that a lot of this is the environment, and that might be why you're seeing this uptick in recent years. But there's also the possibility that there are genetic factors that are predisposing. And so we have exome sequencing data on 3,600 of them, only the cases, but they were sequenced alongside that AMP-T2D study I described initially that had 45,000 patients, including 25,000 controls who do not have diabetes. So we were able to combine these, and through kind of a complicated sample matching process that I won't go into, we're able to arrive at an analysis set of about 3,000 cases and 10,000 controls. So these are 3,000 youth onset cases. And then we did our usual common and rare variant analysis. So fortunately, despite this matching strategy and not having cases and controls ascertained together, we don't see any evidence of artifacts in the associations, which is good. And we do find significant single variant associations. So this is a plot of those. And the ones in blue also are associated with adult onset type 2 diabetes. So there's clearly a big overlap between youth onset T2D and adult onset T2D. Similarly, the rare variant signals also, we found 3 of them, and those also overlap. The ones in blue here are also implicated in either adult onset T2D or monogenic diabetes. And the effect sizes are actually huge. So HNF1 alpha has an odds ratio, I think, of like 1.5 and a p-value of 0.01 in 20,000 cases of adult onset, and is 10 orders of magnitude stronger in youths, which is, it's really interesting how strong these risk factors are. In addition, despite these children not presenting as having monogenic diabetes, about 2% of them appear to actually be monogenic cases. So we then went further and looked at these gene sets again, this time defining them based on HPO terms, which are broad measures of genes based on human phenotypes. And again, we saw evidence of many more associations beyond the significant ones. And actually, you could group them into 3 broad categories. So there's an obesity category, which are genes involved in obesity phenotypes, very different from MODY or monogenic diabetes. Those involved in beta cell phenotypes, which are very similar to monogenic diabetes. And then others that are involved in kind of nondescript measures of hyperglycemia. So multiple pathways potentially implicated. And these are just representative of these gene set associations for one from each of the, you can see that, you know, each gray dot is a gene. It's not like there's a single gene here. Again, evidence of many risk factors spread across these children that are predisposing to this condition. And so we wanted to look further and try to take a more global look of the genetic architecture of this disease. And so since we only had 3 exomite significant genes, we kind of proposed additional tiers of decreasing confidence for genes that could be involved. And just like for our polygenic risk score, we based this on their p-values and also their annotations. And so when this is a kind of a little complicated plot, but let's focus on the red first. The faint circles are the frequency and effect size in adult onset T2D. The dark are the frequency and effect sizes in youth onset T2D, and the size is proportional to the heritability explained. So you can see that they're always shifted upwards. So you're always seeing each of these risk factors has increased prevalence or increased prominence in use. And we did a lot, and there's a lot of confounders here in ascertainment bias and winner's curse that we did really spend a lot of time controlling for. When you then take matched common variants, so just like the strongest common variant signals, you see the same pattern. So the adults have lower, they're more enriched in youth onset cases relative to adult onset cases. And so thinking in terms of heritability, these are our 4 tiers of genes. So you can think of these as just different ways of trying to test the same hypothesis. You can see that the common variant heritability, again, we're trying to match on common versus the rare genes, is higher than the rare. So just like adult onset T2D, the common variants explain more heritability in absolute terms. But if you compare them to the analogous figures in adult onset T2D, they're both higher, okay? And in fact, the rare is proportionately higher than the common are. So just to say that again, a different way, if you look across the population, looks like about 2% of these cases carry monogenic variants. There's a global enrichment for all types of risk factors, but they are 5-fold enriched for rare and 3.4-fold enriched for common relative to adults. So in other words, they skew towards common variants in absolute terms, but rare variants relative to the adult onset form of T2D. Okay, and so that's the population view, but as is the case as earlier, it can hide individual level heterogeneity, right? So for example, the 2.1% of cases with MODY variants look different than the other cases. These are box plots of people with MODY variants versus cases in the same, these are youth cases that do not have MODY. And you can see that they're younger, they're leaner, and they have reduced insulin secretion. That's what this measure is, which fits the MODY phenotype. It's not surprising. So then you go beyond that and look at the people who have high impact or high effect common or rare variants. So this is just an attempt to try to identify people who have high effect variants. And so I have explained in quotes, but we're taking the cases who, when you sum up their rare and common variant scores amongst those tiers of genes we identified, have a relative risk above 5 or above 3. And that's roughly 35%. But don't get too caught up in that, because we're not actually claiming we've explained them. We're just trying to use these to examine patient level heterogeneity. And so this is a plot where now amongst those 35% of cases, you start from those that have the highest fraction due to rare variants on the left. This case is almost entirely has rare variants and very low common variant risk. On the right, you have very low rare variant risk and common variant risk. So clearly those two patients differ significantly, but there's no dividing line between them, right? So there's patients of every type. So it kind of is consistent with that criticism of the McCarthy model that there's no way of bucketing people. But clearly there's heterogeneity, right? There are some cases that are mainly rare, some that are mainly common. Even as a population, there's enrichment for everything, right? So, and in fact, the cases due to rare variants look different than the cases that are dominantly due to common. So the rare variants look a little bit more like MODY and having a younger age of diagnosis, but don't have the BMI and C-peptide associations. Whereas the people who have high common variant risk actually look more like adult onset T2D, where they have it's not significant, but a slightly later age of diagnosis and a very significant increased C-peptide, which suggests insulin resistance. So to kind of conclude, it's kind of interesting. If I had asked you to predict what would be the genetic cause of these cases, you probably would have guessed this, that they would be just have a bunch of everything, and they'd be in between adult onset T2D and neonatal diabetes, because in life, obviously, nothing's ever black and white, and it's unlikely we'll ever be able to bin people fully into one category versus the other. And yet that's how we study rare diseases versus common diseases, where for rare diseases, we're completely non-statistical and we're filtering variants. And for common diseases, we're being these unbiased phenotype, biological hypothesis-free associations. And so I think it would be really cool if instead we could try to be design analyses that for a particular set of phenotypes that describes a patient or describes a pathway, we could build these models to integrate all of the data we have for all of the common traits in the world and map those somehow to these patient phenotypes and leverage this kind of signal, and then enable these open access mechanisms whereby you can type in the gene and maybe a much more detailed phenotype and get these estimates of genetic support. So obviously I'm concluding on a very lofty vision that we'll probably never get to and may not even be possible, but it hopefully gives you a sense of the philosophy our group has and why I pose this question of how we can make genetic data more useful. And if we always are interested in talking to people who are interested in collaborating or joining or just working with us in any way. And I thank you all for listening, and I hope we have at least a couple minutes for any more questions. So thank you all. All right, I see one question that I'll answer in case anybody has any more. How can we integrate other omics type data in this model to help potentially find causation? So I'm assuming that model, that is with regards to this model I proposed here. So I'll just give you one example here to go into a little bit more detail of this. So this model might say that if your gene is involved in disease, the SNP has a distribution of effects that has a higher mean, or actually more accurately, a higher variance, right? And that will depend on the confidence that that SNP is linked to the gene, okay? So clearly, and it also might depend on the SNP's annotation, like is it an open chromatin region in a relative cell type? So you can, but there actually are methods that can estimate the degree to which a SNP's annotation based on all of like the epigenomic data, how much its effect size is increased. And you can also estimate the probability it's linked to a gene based on things like high C, and some of these single cell attack-seq methods that are, you guys have done here. And so those all together is how you can, it's basically in terms of these edges and the distributions between genes and SNPs. Another type of omics data is like gene expression. You can think about like the gene's probability is influenced by its expression pattern. And so that becomes a parameter in your distribution. Of course, you have to fit that, but again, we have thousands of traits and we have tons of genes. And so you actually get a lot of information from genome-wide distribution as to fit those. All right, I see a raised hand, Evan.
Hi, yeah, thanks for sharing all of your work. I'm going to continue asking about this kind of model and how this sort of understanding might work. I think that, as you're saying, there's this kind of question of, are there these modular units that funnel through genes or pathways,
right,
that could quantitatively, like you combine them and you get a score and that puts you in some disease category. But, I think there is just so much kind of in terms of like what to prioritize and how to get to even like testing that kind of idea, because as you say, it feels like it's just forever away, like we might never get there. But since you've, thought about this and looked at all this data, what might you think is the type of data that we can incorporate next? Because I think even the idea that we need to funnel things into genes could be questioned if we believe that there's sort of these other homeostatic method of functions of cells to kind of sequester genes or like stress granules. Like there are like non-genic, properties that could systematically skew kind of like cell state in a way. So I think it's just, there's some like really heavy duty thinking that still remains and what to actually do next.
Right. So, okay. First off, I would say that as a geneticist, obviously we're limited to thinking about what genes can tell you. So anything that you can't detect with genetic variants is not something I'm probably qualified to think about, but clearly is an issue, right? Clearly on the other side, there's clearly a lot that can be gained by simple additive models of like an effect of a variant directly on a phenotype, right? Just to briefly say the type of data, again, we have some very limited internal proof data for this, but I would say that the fact that you now have measurements of a variant or gene's effect across now like 10,000 traits, right? That's incredibly high dimensional data. And now you throw in other things like biomarkers and metabolite, like whatever. There's just interesting, like, can those predict the underlying condition? In some cases it won't, like infection might be caused by multiple pathways, right? But for time and time again, for almost any rare disease we've looked at, like any common disease, the rare diseases that are proxies for it always show up. So the angle I'm mostly interested in as a geneticist is, can we leverage this super high dimensional phenotypic data to somehow map it back? But I don't have enough preliminary data. I mean, I have some on pathway level data, but I don't think I have time to get into that. But that's the best I can do in a minute.
Yeah, no, I appreciate it. Thanks.
I'd be happy to follow up over email. We do have some more thoughts on it. Realize we're at 1 o'clock, so I'm not sure if there's a quick question or whether we should wrap up.
Yeah, well, please join me in thanking Jason Flannick for a great and provocative talk. Thanks, Jason.
Thank you all. And again, I'm happy to answer questions over email. I really appreciate the invite, and have a great day, everyone.